Today, I'm going to talk about DNA-encoded library screening technology uh, and identifying mechanistically distinct inhibitors for antiviral targets. So before I dive into more of our results or studies, I would like to give an overview of how this DNA-encoded library, what is the DNA-encoded library technology and how this works and how this selection process and the library generation, all those things to make it everybody on the same page. So this DNA encoded library technology, or DEL, or DNA encoded chemistry, this kind of emerged in the early 90s. Um, then, and it's kind of more than 3 decades now. It's kind of pretty emerging field throughout the academics as well as industries. And this technology is kind of a merge of a DNA encoding and a combinatorial chemistry approach. So the main principle in this technology is to encode each compound in the library with a unique DNA barcode, and in this, then that enables the screening of a mixture of billions of small molecule compounds together in a single vial. Then at the end, this DNA tag is kind of literally serving as a barcode to identify the identity of a small molecule binder. So there are several advantages with this technology where the first and foremost is the library size can be enormous. You can make billions of compounds. This is definitely an for a chemist or drug discovery community in an expanded chemical space, which is good, right? And coming to the cost wise is much cheaper compared to the other available technology for drug discovery folks in this recent years. Like one of them is a high-throughput screening, and you also don't need to worry about producing a huge amounts of protein. Just as long as you can produce around 200 to 300 micrograms of protein, that's good enough to screen against billions of small molecule compounds. And also, one of the key things that you need to think of before you undertake any drug discovery campaign, you need to how to develop an assay method. How do you want to validate your screen your protein target with along with this chemical matter? In this case, it's just a very straightforward assay method, which is like a affinity-based selection. And there are a huge advancement in these recent years. If you really wanted to, you can fine tune the libraries and refinement curve is available. And we also believe that this technology can be applied towards the targets, so-called undruggable targets. So um, let's say just kind of to further extend, for any efficient discovery process, obviously you have to think of, okay, so you need to have a well-established assay to kind of do a managed high-throughbut screening and development, let's say in case of HTS. And also you definitely need a very fancy infrastructure and high reagent quantities and all those things. And coming to the other technology, like let's say either you need to enable by a structure where you have a clear picture of how to drive this discovery process and further optimization and lead into a lead candidate. And again, it kind of deal with the extensive iterative rounds of measurement chemistry and all those things. And again, as most people think that, okay, and it's kind of slows down the process as if you don't have any of these validated things is not available, and this kind of slows down this drug discovery process. We believe that DNA encoded library technology can bypass all these things. You don't need to worry about having any assay developed in upfront before you start any screening campaign, and also you don't need to have any fancy robots, and you don't need to have a structure. So hopefully by end of this talk, I won't convince you. I think most of these things. So that's well and good with that technology. So how these libraries were made? So I mentioned that we can synthesize billions of small molecules, and how this works, how this process works. I just wanted to kind of give you an idea how this works. So as I said, this is the DNA barcode where uh, a functional group is kind of linked to it through a linker, and where in this case I'm showing a amine functionality, and this is like a reactive handle, like where we attach the small molecules. So in this case, I'm showing an example of using two different building blocks, so having a carboxylic acid blue sphere and a red sphere, And the first thing we're going to do an amidation reaction, capping, then followed by a representing a DNA barcode attachment. So when we do this at this point, we are doing these two reactions in two different separate wells. So after the chemical reaction, we're going to encode with a unique DNA barcode that represents this bullet spear. So after this reaction and followed by the ligation, then we're going to pool at this stage. Both reactions into one well. So in this now we have a well which you have two compounds in there. Then before we do the next reaction, we're going to split equally into two different wells. I'm just kind of an example showing here, two different wells. And again, we're going to do the reaction. Let's say I'm attaching another like a brown spear and a pink spear, and followed by the corresponding encoding with the pink DNA codon. And again, it's two reactions in each well, since we have two compounds in each well separately. Then I pool together, then again split into two different wells. In this, each well is going to be 4 reactants and reacting with the green spear, and the other 4 reactants is reacting with the blue spear. You see this by just doing a 3 chemical transformations of step 1 here, 2 and 3, 2 by 2 by 2, total 8 compounds at the end within the 3 steps. Just imagine if you start from around 100 billion blocks, you're starting in this beginning, then you will end up having 100 by 100 by 100 is like 1 million compounds. So that's the kind of powerful effectiveness of this technology. You can expand the number of the compounds is present in your library enormously within the short number of steps by doing this combinatorial approach. So this is kind of another closer view, a zoomed view of how this DNA, a small molecule attached to DNA. So you see here, like let's say we have done 3 cycles of chemistry, which we have 3 codons, and they're having a linker, and the small molecule is at one point, at one end of the this DNA tag. So if you zoom out, this is a kind of how this headpiece is chemically look like, having a nucleotide base pairs, and the small molecule is covalently attached at this end of the molecule. So by doing this here at Center for Drug Discovery, Baylor College of Medicine, we built around close to I know I mentioned has 7 billion here, so now we're almost close to 8 billion small molecule compounds in our collections. Some of them are 3-cycle libraries and some of them are 2-cycle libraries, which means only there's 2 sequences of chemical reactions. So by doing this, and we kind of cover pretty much most available chemical phase for drug discovery community, and we chose a different type of privileged scaffolds, and we introduced we introduced different chemistries to make the libraries based on those post structures. For example, indoles, piperidines, and biaryls or benzimidazolines, and so on. And we do even synthesize some libraries having a piperidines where we have more of 3-dimensionality into our core structures to kind of explore away from what is available on the commercial market. And also, not only just on focusing on this core structure, we also synthesize some target biased DNA libraries, for example, kinase biased as well as protease biased. So here is kind of another zoomed version of how our library is just an example to showcase like a Marcus structures. You see a triazine, which is kind of a initially an establishment of our thing. Then later piperines and benzimidazolines and so on. So we use our regular practices. We synthesize these libraries in amounts that is sufficient for at least thousands of target compounds. So that's how we operate this. And coming to this is good. So we can make libraries in a huge number, and by maintaining the chemical diversity, then how are we going to screen those? So I'm going to walk you through this. As I mentioned, this screening is a affinity-based selection. I'm going to walk you through step by step how this screening technology works. Let's say once you have this library and we quantify them as a chart of libraries in one Appendorf single tube with billions of compounds, then we're going to incubate with a protein target. So here I'm showing an example, a protein target, the his tag. And this is our regular practice, but this is not necessary to be a hit tag. It can be a flag tag and so on. So once you incubate this protein target with this billions of small molecule collection, then obviously you expect some of the small molecule binds to the protein target, then rest it will be in the solution. So you're gonna pull down by using the tag nickel resin beads, then followed by you're gonna elute them into the solution. So at this point, we're gonna qPCR to determine how the enrichment profile is, how many molecules is burning. Let's say if you happen to have a huge number of molecules, huge DNA present in there, then we're gonna do another round of it's based on the enrichment. We're gonna do 2 to 3 rounds of selections to get the winners of the winners. So once that's done, we're gonna sequence this DNA codons through Illumina next-gen sequencing, and which is gonna give us information of the building blocks or the chemical matter that we included in the process. So by doing this process, we screened a huge variety of target classes, for example, enzymes like kinases, protein kinases, and examples of phosphatases, and nuclear hormone receptors, and so on. And we cover pretty much a different wide span of therapeutic areas, including cardiology, immunology, and oncology, obviously, which is a major thing, and reproductive biology, and infectious diseases as well. So we do have this collection of small molecule libraries, and we do collaborate with the investigator across the USA and internationally. And so far we can screen more than 150 targets in our pipeline. So coming to having this technology and the facilities, so during the pandemic, we thought, okay, let's apply our Dell approach to identify, tackle with this COVID-19 pandemic. I think, so this is kind of an overview of viral life cycle, and which I'm sure most of us know very well by now. I think, in fact, you may be knowing well better than me. So, and once I just wanted to kind of focus on this, like where the highlighted part here. Once this particle get inside the host cell, and that's where the RNA release the viral RNA, and which translates into a polypeptide. And this polypeptide undergoes into a cleavage and to make sure the viral life cycle is going to propagate the viral life cycle. And two of the enzymes that play a very key role in this process, one is main protease and the PL-PRO. And we thought, okay, one of these should be an ideal drug target to work on. And also that historically, you can see the HIV proteases have some success in identifying novel small molecule drugs. So, and also Hep C. So based on that, we chose MPRO as a target, and we screened our DNA-encoded libraries against this around 4 billion compounds at that time. Then we identified several binders. Here I'm showing one of them, and so this is the output that we usually our algorithm works. So where we get the information after post sequencing. So what is the building blocks that you utilized in each round of cycle of chemistry? Let's say in this blue, the cycle 1, red is cycle 2, and this one is 3. So basically, we have created an amide bond here between these two, and the later cross coupling between these two building blocks. So based on this information, we can predict the structure, how the binder that bonds to our protein of interest. So this is the structure, how the heat structure looks like the post selection. You see, this is where the DNA attached, and this is the linker of between the molecule and the DNA tag, and we have a C1, C2, C3, and this is kind of a full length compound. So when we synthesize those compounds of DNA, that means no DNA tag, we try to get the full length compound as well as a combination of C1, C2, and C2, C3. This is just to make sure to understand, okay, which part of the molecule is really necessary in maintaining the activity if it is active. So after getting these compounds synthesized off DNA, we validated by using fluorescent peptide assay biochemically. And you see CDD1724, which is a full length compound, showing around 20 nanomolar of IC50. This is right after the del selection. There's no other medicinal chemistry, just whatever we pull down in the del selection and synthesized with no DNA tag and validated IC50 of 20 nanomolar. That's pretty cool. Again, like coming to the C1 and C2, which is 1712, and unfortunately, this is not active. So, I mean, inactive means we don't see, if we don't see 90% inhibition at 10 micromolar concentration, we call it as inactive. So coming to the other disinthon, which is a C2C3 combination, 1713, it is active at 45 nanomolar, having a 45 nanomolar of IC50. So which is pretty good. It's only twofold difference compared to the parent compound, but we kind of lost quite a lot of mass here. So that's kind of having a good, reasonably low molecular weight and a good drug-like feature. So we chose this compound as our working lead. So as you can see, CDD17 containing aldehyde functional group, which is kind of we all consider as a promiscuous, right? So aldehydes can react any nucleophilic residues in the proteome, and that can be a promiscuous. So without like, let's do some kind of validation here first. How important this aldehyde is. We functionalize like the reduce to alcohol and have generated an amine, secondary amine there, and completely remove the aldehyde. In all these three cases, the compound come dead. So no activity, even at 10 micromolar. So then, okay, that's kind of indicates, okay, this aldehyde is necessary and it is important to maintain the activity. Then next we focused on this individual unit where we just do N-methyl substitution, and that also lead into inactive. So this kind of indicates, okay, this, you need to have this individual NH maintained as well as the aldehyde. So what we thought, okay, let's do what are the other electrophilic. So at this point, we thought, okay, this aldehyde may be interacting covalently. We all know that Emperor has a catalytic cysteine. So we can, to test this hypothesis, we kind of change this to ketone, hydroxymethyl ketone, and nitrile, and alpha beta unsaturated ester. We cannot trap that nucleophilic residue. But unfortunately, most of them are inactive. Only hydroxymethyl ketone shows somewhere 40% inhibition at 1 micromolar. So during the same time, we generated the crystal structure, and it was clear that this aldehyde is forming an adduct with cysteine 145. And also you can see this indazole NH is nicely maintaining 3 hydrogen bonding interactions. So that's kind of nicely correlates to what we have done. But I would not say SIR at this point. This is more of knowing our delicate by truncating the small portions of the molecule. So this is pretty cool. So with these results, we kind of communicated in a quick fashion to PNAS in 2021. So during the same selections, we also identified another structural class. So whereas the other one is a covalent, right? It's an aldehyde having a warhead to interact with cysteine, and this structural class is completely distinct in a non-covalent fashion. So this is the compound, how the compound looks like. So having a benzimidazole chemotype, and this is where the DNA attached. And this is the cycle 2 and cycle 3 building blocks. And so the first thing we observed is having a chiral center in a molecule without a let's synthesize both the isomers. Then we synthesize 1732 having an odd configuration at this center, which is around 800 nanomolar. That's not bad. Right after the del selection, without having doing any work, any follow-up medchem work. So then when we synthesize other NMCM work, then we see around 18 nanomolar activity, which is kind of more than a 45-fold better compared to 1732. So this kind of demonstrate like how important the have made stereochemistry is. Being a chemist, when I see this kind of thing, it feels like great. So okay, you can see the difference between the enantiomer, which is real. So after having that compound, then we quickly tested a few analogs. Okay, what happens if we truncate this part of the molecule, like, which is 1806, and even further truncate by doing this DNA attachment linker and modifying the linker to a cyclobutyl to an open chain structure and so on. So and most of them kind of whatever the modification we made onto the original hit, that did not do any betterment of the compound, but when we maintain this open chain linker, it was around fourfold different, better compared to the parent compound. So whatever is the yellow background are directly after the DEL selection. This is the compound that we identified in the DEL library. And these are the compounds with the green background that we synthesized or we truncated to the deletion. So we have synthesized a few, around 30 more analogs too, but I'm just, for the time's sake, I'm just showing a few of here. Then again, we obtained the crystal structure for this. And you can see this is a distinct class of, it's not like a traditionally classical peptide-like inhibitor, which you can find at least hundreds in the literature from since pandemic time. And this is kind of nicely occupying throughout this different subpockets of this binding pocket and having a distinct mode of binding mode and a binding hash. So with the potent compound along with the other two compounds, we further tested them in an antiviral in a challenging assay in vitro. And these compounds behaved well and showing somewhere two around 3 micromolar IC50s in cell-based viral inhibition assays. Then we thought, okay, how this is pretty good, and we have pretty potency and maintain the viral inhibition as activity as well. How selective are these with the human proteases? So we chose 4 different classes: MMP, renin, thrombin, and cathepsin B, and we tested them against these proteases, and we are delighted to find that none of our identified inhibitors showing any activity, even at 10 micromolar concentration. So which is pretty good, and it is selective towards viral proteases. And we also kind of did a mouse PK and showing TF is not too bad, around one to 2 hours. So next we further tested like how I know these are acting in a different mode of action, binding in a differently compared to the classical peptide-like structures. And we wanted to see when how this behaves different mutants. So you can see in this case, we also compared the nematrolol, which is a drug in the market present. When we compare to a wild type to the other mutations, we see the fold difference is like around 3 to 2 fold different loss of activity compared to these mutants. And if you see this drug, it's like almost 50-fold difference for this delta P168 mutant. And whereas our drugs is like 2-fold difference, 2-fold loss of potency compared to the wild type. I think like this unique mode of action, this improved the resistance profile compared to the nematrolol. That's how we check on it. So I just wanted to kind of give an like just overview how these things like how faster the rate of this hit identification process works. You see from starting to end, it's somewhere like less than 10 weeks, we were in a stage, we had a small molecule inhibitor and validated in a biochemical assay as well as cell-based assay. And a nice lead compound to work on further. So this kind of demonstrated that, okay, DEL can yield to novel chemical matter as a starting points in a quick fashion. So not only that, and this is potent, selective, in a distinct class of inhibitors, not like a peptide-like drugs, which we often find for this proteins. So hopefully, I convinced you all that DEL can be yields a distinct class of SARS-CoV-2 inhibitors and a covalent and non-covalent without dealing much in the selection process, as well as a non-peptidic inhibitors. So we also found that like with a good potency, selectivity, and reasonable drug-like properties. And again, as I mentioned before, this can yield non-covalent and covalent. Both type of inhibitors can be derived from the same selection process, and which showed a favorable inhibition compared to the clinically approved drug nematrolol. Again, the kinetics, which is a big take on, and let's say we have a lot of information for the SARS-CoV, in this case of SARS-CoV-2, from SARS-CoV-1, we have structure and such everything. And that helps us, fortunately, to get the drug into the market in a quick fashion. Just imagine what if we don't have any of those information? It's going to be, takes a lot of effort. So whereas DELS can, without an absence of structural information, you can identify the chemical matter in a quick fashion. So with that, I would like to thank key players and Dr. Yang and Dr. Matsu and Dr. Polskill. And also these are the key players, the work I presented today, Ravi, Shu, and so on. And again, this worked with it's a highly collaborative effort within the Baylor as well as outside of the Baylor. And we are a part of the Midwest AViDD Center. And here I'm showing our group people, it's like a highly collaborative effort. And especially this the clinical variant studies done by Dr. Harris Lab from UT San Antonio. And this is our Baylor team. And I would like to thank the funding, especially we initiated these studies from Baylor internal funds and our U19 NIAID grant and so on. And with that, I'm happy to take any questions there.